In 2015, Lukis Anderson was charged with murder in Silicon Valley. The evidence against him was a DNA match found under the victim's fingernails. The random match probability was 1 in a quintillion. That's 18 zeros. The prosecutor called it ironclad. There was just one problem. On the night of the murder, Lukis Anderson was unconscious in a hospital bed 15 miles away. He had alcohol poisoning. There were medical records, time-stamped entries, nursing staff who saw him there all night. So how did his DNA end up under a dead man's fingernails? The answer comes down to something most people never think about when they hear the phrase DNA match. That phrase sounds absolute. It sounds like proof, but it's not describing what you think it's describing. True Locus examines how forensic evidence is interpreted, presented, and sometimes misunderstood in court. This season explores the language of certainty behind that evidence. If this kind of structured analysis is valuable to you, you may consider subscribing to follow the series. And you're welcome to share where you're watching from. When a forensic scientist takes the stand and says the DNA matches, juries hear certainty. Prosecutors present it as a smoking gun. Defense attorneys struggle to push back. But that word, match, carries assumptions that the science doesn't actually support. It doesn't mean the DNA is identical. It doesn't prove the person was at the scene. And as Lukis Anderson's case demonstrates, it doesn't tell you what happened. Last time we looked at how DNA analysis actually works in a lab. The process isn't fast, it's not automatic. And it takes days or weeks depending on how much biological material you're starting with and how backed up the lab is. But even after all that work, what comes out the other end isn't proof of guilt. It's a statistical estimate. And most people, including most jurors, don't understand that. The common belief is that a DNA match means the crime scene sample and the suspect sample are the same, that the DNA belongs to one specific person and nobody else. You can see why people think that. We've been told for 30 years that DNA is unique, that it's a genetic fingerprint, that it doesn't lie. And your complete genome is unique, all 3 billion base pairs. But here's what almost no one knows. Forensic labs don't look at your genome. They don't sequence your DNA. They're not reading your genetic code. They're counting repeats at 20 locations, that's it. 20 spots where a short sequence stutters. Some people have 7 repeats at a location, some have 12. You inherit one number from each parent, the lab records both. Do that at 20 locations and you've got 40 numbers. A forensic DNA profile is a list of 40 numbers, not a blueprint, not a biological signature, a list that takes up less space than a phone number. When a scientist says the DNA matches, they mean those 40 numbers line up. That's the entire comparison. And the strength of that comparison depends entirely on how rare that particular list is. Some combinations show up a lot, some are rare. To figure out how strong a match is, scientists calculate the odds that a random person would have that same 40-number sequence. They use population databases that track how common each number is at each location, then multiply those frequencies across all 20 spots. The result is what gets read in court. 1 in 10 billion, 1 in a trillion, 1 in a quintillion like in Lukis Anderson's case. But here's what nobody explains to juries. That number, massive as it sounds, doesn't mean what people think it means. It doesn't mean the defendant is the only person on earth with that profile. It doesn't mean there's a 1 in a billion chance they're innocent. And it definitely doesn't mean the DNA proves they committed the crime. What it actually means is narrow and technical. If you grabbed a random person off the street, the chance they'd have this exact same 40-number list is 1 in however many. It's a probability, not an identification. Think about that for a second. The number that sounds like absolute proof is actually just a statement about statistical likelihood in a theoretical scenario. That's a very different thing than saying this person was at the crime scene. So what is DNA evidence if it's not proof someone did something? It's proof that biological material from someone with a matching profile was present. Whether that material connects to the crime is a completely separate question. Forensic scientists understand this. They're trained to distinguish between two concepts, inclusion and identification. Inclusion means you can't rule someone out. Their profile matches, so they're a possible source. Identification means you're saying they are the source definitively. In court, scientists testify to inclusion. Juries hear identification. And that gap is where wrongful convictions happen. Here's the kind of scenario that plays out in courtrooms constantly. Someone breaks into a house, smashes a window, steals valuables. Police recover blood from the broken glass. Weeks later, they arrest a suspect. His profile matches. The forensic scientist testifies carefully. The defendant cannot be excluded as a possible source of this DNA. The probability of a random match is 1 in 5 billion. What the jury hears is simpler and far more damning. His blood. On the glass. He was in the house, he did it, but the scientist never said that. She said the profile matches. She didn't say how the blood got there. She didn't say when. She didn't say the defendant broke the window or committed the burglary. DNA can tell you who. It cannot tell you how, when, or why. And that distinction matters more than most people realize. If the defendant visited that house 2 weeks earlier to fix the plumbing and cut his hand on a tool, his DNA would be there. The match would hold. The statistic would be identical. 1 in 5 billion. The science would be accurate. The conclusion would be wrong. That's why context matters. Witness testimony, timelines, other physical evidence, motive, opportunity. The DNA profile is one data point. It doesn't interpret itself. And here's something else most juries never hear. The statistics assume the person you're comparing isn't related to the actual source. That assumption is baked into every random match probability calculation. But what happens when it's wrong? What if the suspect has a brother? Siblings share DNA, not all of it, but a lot more than random strangers. At the 20 locations forensic labs test, brothers can have very similar profiles, usually not identical but close. And when profiles are close, the statistics collapse. That 1 in 10 billion number, it can drop to 1 in 10,000 when you're comparing siblings. Sometimes lower, the match is still there, the science is still valid, but the strength of the evidence just fell off a cliff, and nobody tells the jury that this calculation assumed the suspect and the real source weren't related. That assumption gets presented as if it's a fact. It's not. It's a condition. And if the condition doesn't hold, the number means something completely different. So when a forensic scientist says 1 in a billion, what they're actually saying is this: assuming the source isn't related to the defendant, assuming the database accurately represents the population, assuming no errors occurred during collection, testing, or analysis, assuming those 40 numbers actually came from one person and not a mixed sample. Then the chance of a coincidental match is 1 in a billion. And that's not one assumption, that's 4. And every one of them can be wrong. Most jurors never hear this spelled out. They hear the number 1 in a billion, 1 in a quintillion, and they hear it as certainty, as proof, as something you can't argue with. Defense attorneys can push back. They can bring in their own experts to challenge the statistics, but that takes money and time. Most defendants don't have either. And even when the challenge gets made, it rarely lands. The number is too big, it sounds too scientific, it drowns out everything else. Then there's what happens when the sample isn't clean. Not every crime scene gives you a perfect DNA profile. Sometimes the sample is tiny. Sometimes it's degraded. Sometimes it's been sitting in the rain or the heat for days. In those cases, the lab can only generate a partial profile. 15 locations instead of 20, maybe 10. Sometimes fewer. The weaker the profile, the weaker the statistic. 1 in a billion becomes 1 in 100,000, or 1 in 10,000. In bad cases, 1 in 100, that's still called a match, but it's nowhere near as strong. And then you have mixed samples. DNA from 2 or more people in the same evidence, blood from multiple people, skin cells from a door handle that 10 people touched. Separating those profiles is complicated and subjective. Analysts use software to estimate who contributed what, but the software requires judgment calls. Which peaks belong to which person? How much DNA did each contributor leave behind? Was this deposited all at once or over days? Different analysts can look at the same mixture and reach different conclusions depending on the software they use and how they interpret the data. The testimony in court will still cite probabilities. It'll still sound definitive. But underneath, there's interpretation, assumption, judgment. This doesn't mean DNA evidence is unreliable. It's not. When it's used correctly, with proper context and corroboration, it's one of the most powerful tools in forensic science. But it does mean DNA evidence doesn't speak for itself. It needs context, it needs investigation, and it needs to be understood for what it actually is. Which brings us back to Lukis Anderson. The DNA under the victim's fingernails was real. The match was real. 1 in a quintillion, But Lukis Anderson was 15 miles away in a hospital bed when the murder happened. So, how did his DNA get there? The answer turned out to be transfer, indirect transfer. Hours before the murder, paramedics responded to a call about Anderson. He was unconscious from alcohol poisoning. They checked his vitals, touched his skin, then transported him to the hospital. Later that same night, those same paramedics responded to the murder scene. They tried to revive the victim, and in the process, they transferred trace amounts of Anderson's DNA from their equipment or their hands to the victim's body. Nobody contaminated anything on purpose. Nobody made an obvious mistake. The DNA evidence was scientifically accurate. Anderson's profile was under the victim's fingernails, 1 in a quintillion. But the conclusion that he committed the murder was wrong, because the DNA couldn't tell anyone how it got there, or when, or why. Anderson spent 5 months in jail before investigators figured out what happened. If the medical records hadn't been so clear, if his lawyers hadn't pushed, if the timeline hadn't been ironclad, he might still be there. The DNA match was never in dispute. The interpretation was, a match means the profile is consistent with the sample. It's a list of 40 numbers that line up. That's all it means. It doesn't prove guilt. It doesn't establish presence. It doesn't explain how, when, or why DNA ended up where it did. Those are investigative questions. And they require more than a lab result to answer. The phrase DNA match carries so much weight in a courtroom that it can overpower everything else. It sounds absolute. It sounds like the kind of evidence you can't argue with. But what it actually represents is conditional, probabilistic, and dependent on context that juries almost never hear. Forensic scientists know this. When they testify, they're careful. They say, cannot be excluded. They cite probabilities with conditions attached. They avoid saying someone was definitely at a scene. That caution is deliberate. The science supports the comparison. It doesn't support conclusions about guilt, but that nuance gets lost in translation. Juries hear certainty. Prosecutors argue certainty.